Hello, everyone. My name is Dr. Anna Zelaya, and today I'm going to be walking you through how to analyze your next generation 16S data using DNA Subway.
Now, I tried to keep this tutorial as brief as I possibly could. I really just wanted to go over the most important things that you needed to know to complete your analysis.
So, the first thing you'll want to do is log onto DNA Subway, and you'll want to do that by going to dnasubway.cyverse.org. And that will take you to this homepage here. Now, in the top left corner, you can enter your username and password. Now, this should be the same username and password that you used when you created your Cyverse accounts and you uploaded your FASTQ and metadata files into your data store. So that same username and password that you used for those steps is what you're going to use to log into DNA Subway. So go ahead and type those in and then click Login. And then that will take you to this page here where any projects that you may have started will be listed. Now, if this is your first analysis, you will likely not have any projects. This will be blank. I have a few projects here that I have previously analyzed. But you'll want to start a new project. And the way that you do that is you click on this purple box here that says microbiome analysis to create a new project. And that will take you to this page here. And so we're going to select a few things here on this step to start our project. For the project type, you're going to click on paired end reads. For the file format, you will click on Illumina Cassava 1.8. Next, we're going to go ahead and give our project a title. You can name your project anything you would like. I will go ahead and just type tutorial. You can type in your group name, your class name, the semester that you're working with anything that would make sense to you for your title. And for the description, you can type, what I would recommend you type is something that describes the experiment that you did. For example, the sample data set that I will be working with today was done on beetles that were fed different bean diets. So I will go ahead and write something like 16S microbiome analysis of beetles fed on different diets. So you can type anything here that adequately describes your experiment. And then once you've filled in all of the fields, you'll go ahead and click on continue, and that will take you to this homepage here. This is the main homepage with all of the steps that are going to be performed in our analysis. You'll notice that there's three main steps with each one having a few different sub-processes. So the first step is metadata and QC, which has three main sub-processes. Then we have clustering sequences, which has one main subset, and then alpha beta diversity, which has 5 sub-processes. We're going to go ahead and ignore these last steps for now. We won't be doing that in this analysis. You'll also notice that many of these have this red X next to them. That means that we can't really proceed to those steps yet. We're not ready for them. So this green R means that pretty much that's the only thing we can do right now. So that's where we're going to start. So we're going to go ahead and click on Manage Data. That opens up this next window here, which will allow us to upload our files for analysis. So we'll go ahead and click on Add from Cyverse. And this is going to take you to, it's going to give you a few options. If you uploaded your files into your folder in the data store, you will see a folder here that has your username on it. So mine says Azalea, that's my username. So I'm going to go ahead and click on that. You should click on your username, the folder with your username. And then I have my files in this folder called Beetle Diet Microbiome. So wherever you stored your files, go ahead and click on that folder. And now I see all of the files that I will be analyzing. Now I'm going to go ahead and add all of the FASTQ files in this directory. So I'm going to click this button. If you have some samples that you don't want to analyze for some reason, you can manually click on each file that you do want to analyze and then click on Add Selected Files. But I want to analyze all of my files. So I'm going to go ahead and just click on Add All FASTQ Files in this directory.
So alternatively, if your instructor is sharing the data with you, then you will want to access the files a different way. So you would go to this folder right here that's called Shared with Me, and you would click on that, and you'll see this text box that pops up, and you'll want to write in either the email address of your instructor or the username of the instructor. This information should be given to you by your instructor. And so if your instructor, for example, has the username of L Bloomer, um, you can type that in and then click on this search word. And then you'll see some additional information that pops up. In this case, it's the name of the instructor and the email address. Go ahead and click on their name that popped up. And then you'll start to see basically all the folders that are in their folder, other files that are in their folder. And so make sure that you have the name of the folder that you need from your instructor. In this case, I'm going to click on demo data, which is going to go to some more files. I'm going to click on this first one here. And then that is what's going to show me all of the files that I need to run my analysis. And so I'll do the same thing that I did previously. I'm going to click on this button here that says add all FASTQ files in the directory to add them to DNA Subway.
So now that we've added all of our FASTQ files, there is one more file that we are going to need to run DNA Subway, and that is called a metadata file. We won't need to worry too much about creating this file or uploading this file because DNA Subway actually just creates it for us, which is really nice. So if you go down to the metadata file section and go to create new, it takes it a few seconds to create this file, but once it's created, essentially what you're going to see is a spreadsheet with 2 columns in it. Here we go. It has 2 columns in it, and basically it lists out all of the different samples. You don't need to worry too much about this file for now. We will actually be making our own later on when we go over the R-Shiny tutorial, but for now, DNA Subway just makes it for us, which is really nice. So now that we have it there, we check that all of our samples are the right samples, everything looks good. We're going to go ahead and save that, and then we'll see there that it says metadata file created. And then you go ahead and click back to get back to this screen. So we have our FASTQ files, we have our metadata file, and now we just need to validate this metadata file. It's just to make sure that the formatting and everything is correct, which it is. There shouldn't be any warning signs or anything here. It all looks good. So now you can go ahead and click run.
And now it takes me back to the kind of main process page or the home page, and I'm ready to go to the next step, which is the demultiplex step. Now in this step, you'll see this window here. It says random sequences to sample for QC. QC means quality check. So it's basically going to take 10,000 random sequences from all of our files and use them for the quality checking that we'll describe. I'll describe in a little bit. But for now, let's go ahead and just hit this demux reads button. Now this step is going to likely take quite a while. It will take anywhere from 5 minutes to, it could even take up to an hour. As you can see here, it says this job, it gives it a job number. So my unique job number is 36589, and it says that it's currently running and that as soon as it's finished, it will display the steps here. Now at this point, if you don't want to wait for it to run, you can go ahead and X this out. You can even close out this browser. You can go have lunch, go have coffee. You can watch other things or do other work. You can even close down your computer. It will still keep running in the background. This step does take a long time, so you might want to just go ahead and kind of work on other things or take a break. So a quick description about what the demultiplex step is doing. It's basically taking all of our files. Each file represents a sample, and each file contains thousands of sequences that represent different potential bacteria. And it's basically taking all of those sequences and analyzing them all together and keeping track of which sequences come from which samples. Another thing that it's doing is it's performing a quality check on each of those sequences, which will allow us to be able to throw out poor quality sequences or low quality sequences and only keep the high quality sequences for further analysis. So the results of this step will allow us to determine which sequences we can keep and which we can throw out. So if you decide to take a break during the demultiplex step, just make sure to check on it periodically to check on the status of it and check to see when it's done. When it is finally done, you will see these two symbols next to the step, these next two steps. So there'll be the white V in a green background, and that means that there is results there that are ready to view. And then the next step will have the green R and a white circle, which would mean that you're ready now to go to the next step. But before we go to that next step, we're going to want to view the results of the demultiplex step. So when you click there, you'll see this window, and you'll want to go to here where it says results, and there's a demultiplexing summary. Go ahead and click on that. And this will then take us to the QIIME 2 viewer, which will show us the results of this step. So to better see these results, you can go here to the maximize window button and click on that, and it'll just open things up and we'll see everything better. And you'll see that there are two tabs in this viewer. There's an overview tab and an interactive quality plot tab. The overview tab just gives you some basic kind of statistics of all of the files and all of the sequences that were in there. You can scroll down and see the number of sequences that were in each of our samples. My sample data set has 6 samples, and here we see how many sequences were in each one. But what I really want to see, what I really need for the next step, is found here in this interactive quality plot tab. So we click on that, and what we see are 2 plots that are interactive, and I'll show how you can interact with them in a minute. But these tabs allow us to assess the overall quality of the sequences that were demultiplexed. Now there's a lot going on here, so we're just going to walk through these slowly. Let's focus here on this first plot, which is labeled forward reads. Forward reads, here the terminology that's being used is reads instead of sequences. Reads means the same thing as sequences. So here what we mean is the forward sequences or the forward reads of our data. And you'll notice the axis of the plot. So the x-axis is labeled sequence base and the y-axis is labeled quality score. And so essentially what is being shown here is the quality score of each nucleotide base along our sequences. And this is the average of the 10,000 randomly subsampled sequences. If you remember at the demultiplex step, it said 10,000, it was going to subsample 10,000 sequences for quality check. Well, now these are the averages of the quality scores of each of the nucleotide positions of those 10,000 sequences. Now the x-axis ranges from 0 to a little over 220 for the forward reads. What that means is that for the subset of 10,000 sequences, the maximum sequence length observed was about 220 base pairs, which is what we would expect given the type of sequencing that we did and the V4 region that we chose for sequencing. Now you'll notice that the y-axis ranges from 0 to 45, and those are the range of quality scores that each base position could have. So in general, a quality score above 30 is considered high quality, meaning that you can be highly confident that the nucleotide base was called correctly during sequencing. Scores above 25 are considered good, acceptable, and sometimes even scores around 20 are also considered acceptable. But anything below 20 is typically considered a low quality base call, meaning that you can't be confident that it was called correctly during sequencing. And if we can't be confident about that, then we really don't want to keep those bases when we continue with our analysis.
So the goal of this step is to find the bases that are of low quality.
And then we're basically going to remove them or trim them off. That's the terminology that's used during sequence analysis. We're going to basically trim off
Any bases
that are not good quality, and we'll proceed with sequences with only the high quality portions of our sequences for the further steps in the analysis. So let's take a closer look at our forward reads. So it's the process that we're going to do is really simple. I'm going to scroll down a little bit so that we can take a look at this parametric 7-number summary table that you see down here. These are basically statistics of the box plot charts that we're going to want to pay attention to. So what I'm going to do is basically put my mouse over here in the box plot of position number 1, and I'm going to click right there. Now what you'll notice is that the parametric 7-number summary here starts now at position 1 because I clicked on there. And if I move along, the bases, then this position moves along with me, and all of the stats of each position also changes. So if you move along and you check on that table, you'll see that the numbers kind of change. So that's what we want to do. We're going to start from the very beginning and take a look at this here that says bottom of box 25th percentile and the quality score associated with the 25th percentile. That's what we're going to be paying attention to. And we're going to look to see when this value drops below 30. So I'm going to start here at the very beginning and kind of move along, and I'm noticing that the numbers are staying at 40, dropped a little to 38 there. That's okay. It's dropping, going between 30 and 40. Let's just keep going along. All of these values are looking good. You'll see other values changing, but we're not paying attention to those. We just want to focus on 25th percentile. And what you'll notice is that it doesn't really change until the very last base, which is at position 224. And you'll see that at that position, this value is at 13. So this is where we're going to trim off our forward reads, is at position 224. So what I would recommend is that you write down, you do this same procedure for your data, and you write down the position at which this value drops below 30. Then you're going to go ahead and do the same exact thing for the reverse reads. So when I do that for the reverse reads, we're moving along, and we see that the value drops at position 231. So that's the value that we're going to write down for our reverse reads, is where we're going to trim. Now, one thing about this selection process is that it is a kind of recommendation. It is not always the case that your data is going to look, in fact, your data most certainly will not look like this data that you see, like my data. Every sequencing run is different. And it's possible that you will see nucleotide bases drop really, really early on. It's possible that within the first 100 bases, you might have 1 or 2 or even 3 positions that are really, really low, and then they kind of go back up again and they're high. If that happens, in general, you wouldn't want to trim too short. If you have very low quality bases below, position, maybe I would say maybe 150 or 160, you don't really want to trim that low, because the further analysis steps will not work properly. So you really don't want to do that. What I would recommend, if you have 2 or 3 base pairs that are really low in the beginning, maybe just ignore those and focus on the ones that are at the end, anything like about above 160 or 170. These are the ones, if you start to see them drop here, then you can go ahead and pick there to trim. Now, if you have a lot of bases that are really, really low, below 25, below 20, really early on, if like virtually every single one of them is low, then that's an indication of a bigger problem. That's an indication that your sequencing run probably was not very successful. You have very low quality sequences in a lot of your samples, and you probably won't want to continue with analysis if they're that low quality. So if that happens, feel free to reach out to me. We can discuss, strategies for what to do in that situation. Hopefully that's not the case for you. But if you do require, if you have any questions about where you should trim, I'm happy to answer any questions to help help you analyze your data. So once you have recorded the position for the forward and reverse reads where you're going to trim, then you are ready to proceed to the next step. So you can click on this back button here, and we can just go ahead and exit out of here and then go back to this front page and click on the next step, which is data 2. Okay, so at this step now, we're going to take the parameters that we got from the previous step, the previous demultiplexing step, and input them into the data 2 step. So you'll see here there are a few boxes, the forward and reverse. There's trim left for both forward and reverse. And we're going to go ahead and ignore those for now. We're going to move on to the trunk length for the forward reads, which if you remember from the previous step, the value that we were going to put in here was 224. And then for the reverse, the value that we were going to use there was 231. And so, of course, the values that you input here will not be these numbers. They're going to be the numbers that you determine to be the best trimming parameters for your data set. I just realized that I've been using the terminology trimming for removing of low quality base pairs of our sequences. But then there is this kind of trim left section that we just kind of ignored. So I just want to talk a little bit about that. Trimming is just a general term to remove low quality base from our sequences. But in data 2, there are some definitions down here, the trim left and trunk length. You can go ahead and read those, but basically data 2 refers to trimming as removing bases from the beginning or the 5' end of the sequences, which we didn't do, and that's why I ignored it. And then the trunk length terminology is what they use for removing base from the end of the reads, and that's what we did. So that's why we're filling in those boxes. So go ahead and feel free to read those definitions to get a better understanding about the terminology used in data 2. But once you're done with that, go ahead and press the Trim Reads button, and that will start the process. Now, this process, the data 2 step, is going to very likely be the longest step in the analysis. I've had it take several hours. I've had it take overnight. Depending on the number of samples that are running and the number of users on DNA Subway, it could take, the shortest could be maybe about an hour, and it could take several hours to even an entire day to run. So what I would recommend is just, after you start this step, go ahead and close your browser, work on other things. Maybe don't check on it until the following day. If it's in the beginning of the day that you start it, go ahead and check it, maybe mid-afternoon or even nighttime. If it's at night when you start it, go ahead and check it in the morning. But just give it some time to run, especially if your entire class is going to be working on this at the same time. It is likely that it will take a long time. So just check on it every now and then, but don't be surprised if it takes a long time. And I can explain a little bit about what Data 2 is doing here. So Data 2 is a program that is used within QIIME to essentially quality filter out all the data. So in the previous step, I'm going to close out of this right here. So in the previous step, in the demultiplex step, we basically took our sequences, we sorted them and ordered them based on the samples that they came from, and we looked to see what their quality was based on the quality scores that are provided during the sequencing run. We took that quality data, we said, this is where we want to trim or truncate our reads. And now Dada2 is actually doing the truncating and the trimming. There's a lot of steps going on in this process, that a lot of details that you don't necessarily need to know at this point, but this is why it takes such a long time. And so as we can see here, there's this R, the red R in the yellow circle, the process is running. So go ahead and just close out. And when you come back, you should see the white V with the green circle, and that will mean that the process has stopped and that the data is ready to be viewed. Okay, so at this step, Dada2, it's been a couple of hours and Dada2 is now finished. So we can click here into Dada2, and you'll see here that there is a section here that shows the results of Dada2. Now there's a lot of good information here, but for the purpose of our tutorial, we don't need to know all of that information to get the CSV table that we need. Really, the only information that we need is found here in the stats link. So I'm going to go ahead and click on stats, and I'm going to maximize this here so we can see better. And what you'll see is this table. Now, this table basically provides, it shows here all of our samples that were filtered using Dada2. It shows the number of sequences that were in each of the samples when we started Dada2. And then it shows the number of sequences that basically survived the filtering steps along the way in Dada2. So there's 4 filtering steps in Dada2. And as you can see, as we move along this table, there's kind of less and less sequences as we move along to the very end, to this final filtering step. So essentially, this column here gives us the end result of the number of sequences that we have left after Dada2 filtering. And so the information that we need here from this is you want to look at all of your samples and you want to record the value that is the smallest value. So out of all of ours here, we see that the smallest value here is 90,417. That's associated with this sample here, ADZU3. So we're just going to write that number down, 90417. Go ahead and look at your data, find the sample with the fewest sequences left. Now, again, this is another thing where if you need some help determining which is the right thing to do, please email me. I say that because sometimes some samples there was maybe a problem in their sequencing where a lot of the sequences that were in that sample, they just didn't survive the filtering process. So if this number here would have been 1,000, I probably wouldn't want to record that value. I'd actually record the second highest value or the second lowest value, if that makes sense. So it's fine that we're losing sequences along the way, that's normal, but when we lose too many sequences, that's an indication that there was a problem with that particular sample or there was a problem with sequencing. So I guess maybe as a general rule of thumb, you could say if your smallest sample is about 10,000 sequences or more, go ahead and keep that. But I hesitate to even give you that value because it really is a case-by-case basis. It really depends on what your input sequences were and how many sequences you started off with and how many you ended up with at the end of it. And so if you're losing the majority of your sequences, if you're losing 80 and 90% of your sequences, that's usually not a good sign.
You know, but if you're losing 10%, 20%, even maybe 30%, sometimes that can be okay. So all of this is to say, if you look at your data set and you are not sure if the number of sequences that you have at the end of data2 filtering is a reasonable amount, please feel free to email me. And you can also download this table as a TSV file if you click on this link here. And so if you download it and you're not sure and you want me to take a look, you can email that table to me, and I can look over your data and give you some recommendations about what I would choose if it was my data set. For now, for this data, I'm going to go ahead and choose this number. So I'm going to X out of this box here. And that's really the only value that we need for this next step. So we're going to go next to alpha rarefaction. And for this step, we're going to, for this maximum rarefaction depth, this is where we're going to input that value. So I'm going to put in 90417. You're going to input the value that you chose for your data. And then I'm going to hit submit job. Now this step might also take a while. This is kind of a variable step for me. Sometimes it happens really fast. It's done in like 5 minutes, and sometimes it takes over an hour. So it's hard for me to gauge how long this step might take you, but do plan on it being several minutes and maybe even longer, especially because it's very likely that you know, a lot of users are going to be on at the same time from your class. So maybe give it an hour, check on it every hour or so. Now, what is the alpha rarefaction step doing? I think it might be easier if I explain it when it's finished processing. So go ahead and pause here and we can start up again once the process is completed. Okay, so the alpha rarefaction step is completed. This is a Sunday night. Probably not a lot of people are using it right now because it was over in about 5 minutes. But I also realized that this step, you will be seeing alpha rarefaction again when you do the Rana Kappa tutorial. So I think for the sake of, okay, so the alpha rarefaction step is over now. And so to move on to the next steps, we are going to just take a quick look at the results of the alpha rarefaction step. And I can explain a little bit about what it's doing. So I clicked on the button, and now I'm going to click here on the link alpha rarefaction plot. I'm going to maximize this to take a look at what we're looking at here. And if you scroll down, you're going to see two plots. Let's go ahead and just focus on the top plot for now. The bottom one, we're not really going to pay attention to that plot. And you'll see here that there's two kind of fields here to be selected. So there's the metric, and if you click on that, it'll say Shannon, Faith, and Observed OTUs. So these metrics are basically diversity metrics. They give you an indication of how diverse your samples are, how the different types of bacteria that are in the different samples and how diverse they are. So just to kind of briefly go over this and give you the most important information that you need to proceed with this tutorial, click on the Observed OTUs metric, and your sample, the sample metadata column, you might have different options here. In my metadata file, I had one column that was called host, and that represents the bean that was fed to the beetles. So I had 3 different beans, and if you look over here, my chart says Adzuki, BEP, and Hyacinth. These are the 3 kinds of beans or hosts. So even this legend here is going to be specific to your data. If whatever your experiment was, whatever the different parameters or treatments were, that's probably what you're going to see here in the different colors. So I'm going to go ahead and leave host for now. And so now let's take a look at this plot here. And so the x-axis is labeled as sequencing depth,
The y-axis is the observed OTUs, which is the metric that we chose. So essentially, what this plot is showing is the number of observed OTUs, or in this case, you can think of an OTU as it's kind of like a bacterial species, although it's not exactly like that, but you can, for the sake of our tutorial, we're going to just kind of call it that. But it's the number of unique types of bacteria that are found per sample for any given number of sequences that were randomly subsampled. So essentially what rarefaction does is this mathematical process that takes all of the sequences from all of the samples, randomly draws a sequence, kind of keeps track of which samples that sequence was found in, and then it kind of stores it in memory. And then it takes another sequence, randomly draws out another sequence, and then it compares it to that first sequence. And if they are identical, then that means that they're probably going to be classified as the same type of bacteria. So that's still, even though there's two sequences, that's just one bacterial type. Then it'll take a third sequence, it'll compare it to those, and if it's different, if it's not 100% identical, if it's different, then it says, okay, this is probably a different kind of bacteria. So after pulling 3 sequences, I have 2 different kinds of bacteria. And then it just keeps doing that. It pulls out a 4th sequence and a 5th and a 6th, etc. And it keeps track of how many of those were the same, how many were different. And in the end, you have the number of observed OTUs, or the number of unique bacterial types from that process. By continuing to do that over thousands and thousands of sequences and keeping track of which unique bacterial type is found, you can start to get plots like this. So here, after 10,000 randomly sampled sequences across these different samples, in this dark blue here is Adzuki beans. So in Adzuki beans, after randomly drawing out 10,000 sequences, there were about 25 unique bacteria that were found in that sample. So basically what that means is that even though there were 10,000 sequences, they all represented only about 25 bacteria. For hyacinth, that number is a little higher. It's maybe about 28. And for black-eyed pea, it's the highest. It's 50. So there was a lot of redundancy here. Basically, a lot of these sequences ended up classifying as the same type of bacteria. And that's all we can really say right now. We don't know what type of bacteria they classified as, but there's a lot of redundancy here. And then if we go and now you repeat that process, but you take 20,000 instead, you start to notice that these curves start to plateau. They start to plateau here, they start to plateau here, kind of going that way. So what we're looking for, we are looking for this kind of plateau, because basically what that means is that after about 20,000 sequences, it doesn't really matter how many more sequences we have. We're not really finding any new bacterial types. We kind of found everything that there was. We found all the diversity essentially from each of these samples after about 20,000. And then we just kind of started picking the same bacterial types again. For now, I wanted you to see this because this value of 20,000 is what we're going to remember for the next step. So for your data set, what I'd like you to look at is, where do you start to see your curves start to plateau? If you see a straight line just going all the way and never ending, and it's just straight, then that's not a good sign. Basically what that means is that your samples are really, really highly diverse. They're so diverse that even after sampling 50, 60, 70, 80,000
sequences, you're still finding new and unique types of bacteria. So based on the extractions that you did and the sequences that you got back from that, you didn't get enough sequences to fully truly capture the diversity of your samples. You're underestimating, or you have an underrepresentation of what truly existed in your samples. But hopefully you do have charts that start to plateau at some point. And so basically what I'd like you to do is just find that value where they start to plateau. For my data, I am going to choose the value of 20,000. You could argue that it starts to plateau at about 10,000, but I'm going to go ahead and choose 20,000 based on the black-eyed pea curve. So once you determine that value, so I'm gonna grab this number of 20,000. That's the number I'm gonna record for the alpha rarefaction step. So now I'm gonna click out of here, and I'm gonna move on now to my core metrics step. And so here now, this is sampling depth. That's gonna be based on the value that we just looked at in the alpha rarefaction. So I'm gonna choose 20,000 here. And actually, just to let you know, if your value is higher than 20,000, if you saw your sample start to plateau around 30,000 or 40,000, unfortunately, there's a limit to, I don't know if it's specific to DNA Subway, but there's a limit to the value that you can input here. And 20,000 is the maximum value. So if your value is higher than that, go ahead and just put in 20,000, because that's the only value we can put. If it's lower though, it would be best for you to pick the value that is closest to your data set. So if you saw your data start to plateau around 10,000, go ahead and put 10,000 at this step. For this next section here, we're gonna choose our taxonomic classifier. So this is gonna be essentially the database that QIIME will reference to assign taxonomy to our bacteria. So we're going to know what phylum they are, what phylum, class, order, family, all of that taxonomic information. We're going to be able to assign that based on the databases here that we find, or that we choose. So I'm going to choose here this Green Genes 515F806R. That is also what you want to choose, because this is specific to the primers that we used when we sent off our samples when you sent off your samples for sequencing, and this is what you chose. So that's what you're going to pick, and then go ahead and submit job. And this step might take a few minutes. Again, it might take up to maybe an hour. So just keep checking on it periodically until it is finished. Okay, so the core metrics step is done. It took about 20 minutes. And now we're coming towards the end of our analysis. There's not anything really here to view at this point. I mean, there is things that you can view, but it's nothing that we really need to finish things up. And here at Alpha Diversity and Beta Diversity, these two tabs do have some good information, but you're gonna do these steps later in a tutorial with another online teaching tool. What we're going to do instead is we're going to go to this taxonomic diversity tab. I'm going to click on that, and you're going to see here the results. There's these two, there's bar plots and taxonomy. We're going to go to this bar plots right here. I'm going to maximize this window, and there's a few things to look at here, but basically the most important one is this taxonomic level. So level 1 is, so in the previous step, we classified all of our sequences using that Green Genes reference database. And so level 1 is what's the domain. Here it has a K here for kingdom, but it's also the domain. So basically bacteria, archaea, eukarya, those are the 3 domains of life. And so that is what the level 1 is classifying at. So what you see here on this chart is that all of our samples had bacteria as the only domain
that was sequenced, which is good. That's what we wanted. That's the primers that we chose. So that's exactly what we wanted. For your data, you may also see archaea here. That's fine because the primers that we chose, the 515, 806 primers, those are also going to select for archaea. You might also see eukarya here. And that's also fine. Sometimes that happens because, you know, we're dealing with beetles, which are eukaryotes. They also might have some bean matter still in their guts. So when you extracted DNA, you might have also extracted some bean DNA as well. And so it's okay if you also see eukarya here. And in future steps outside of DNA Subway, you're going to be filtering all of that out anyway. So it's fine if you see all three domains there. Level 2 is phylum. And so what you see here is in this bar chart is all of the bacterial phyla that were present in all of my samples. Level 3 is class. Level 4 is order. And level 5 is family. And this is what we're gonna download the data for. We want to know at the family level what is the diversity of all of our different samples. You can also kind of change the color palettes here if you want to. I'm just going to ignore that for now. And then here, this just kind of helps you sort how you want to look at the data. So if we put host, it just kind of changes the order here of the bar plots. So you can do that if you want it in a certain order. And then here, this is where you're going to download. So the file that you need for further community analysis is this CSV file. So if you click on this CSV file, it's going to download a CSV file into your downloads folder. You can then save that into a folder where you're going to be able to access it later when you do the rest of your community analysis. You can also download this chart if you'd like. It makes a nice chart for presentations. And so you can kind of click on this bars. It downloads the two different parts. The bars will be the actual bar chart, and the SVG legend is going to be then the names of the bacteria. So that is it. The most important thing here is you make sure you download that CSV at the level 5 taxonomic level. That's what you need for this, for DNA Subway. So that marks the end of our tutorial. We walked through how to import all of your data into DNA Subway via the data store. We then demultiplexed that data and were able to look at the quality of that data and then filter low quality sequences out of our data set. So what we ended up with was a taxonomic, a CSV file with high quality sequences that were classified at the family taxonomic level. So this is the end of our tutorial. I hope that this was helpful to you.